Next, let's go to Browse and Study page. In the study page, we can see all experiments or studies of the current crop. Each such study contains traits data measured on genetic lines or germplasms that we viewed before. Once we select one of the studies, several tabs appear, and on the right we can see traits that were measured in that study. The Observations tab shows all data. Each row represents an observation made on a particular line. So, we can see the line name, as is typically written on the seed's envelope. Then there's the breeder name, which is the name recognized by the breeder. Next, there's the generation of that line, and all other columns represent traits data. We can click on the sign near a column's header to see more details about this trait. Here, for example, we can see that this column represents plant habit, and these are the possible values that it can get. This column stands for fruit weight, and the scale is grams. Users with appropriate privileges can edit the table just like this. Note that if by mistake you enter an invalid value, the system will identify it and alert you accordingly. Also, you can download the table as an Excel file. The table can be split by column. So you can scroll to the right and edit values while viewing line names from the left. Another option is to hide columns. So here I'll just take several columns, drag them to the right, and now they do not appear anymore in the table. The filter button is used to search for lines according to defined criteria. Here, if I only want to see lines with a plant habit of 3 and where fruit size is at least 4, only lines that match these criteria appear now. Next, there's the Description tab. They can be used to set general info about the study, like this, such as name, title, description, and dates. And finally, Study Query shows all studies as a searchable list. Under the Trait menu, there's a list of all traits in the system. I can search for a trait according to name or by the traits group. Let's select all traits that are fruit related. I can click on a trait and enter into its details. Here I can see all studies in which this trait was measured. Clicking on this button will change the display. This shows the scale and method used to measure the trait. On the bottom there are all possible values for this scale, and this can be edited if necessary. This is the end of this demo that shows data browsing and general interface of Phenom-Networks. Other demos will cover the breeding process, which is the operative daily use of breeders to set up an observation field and create crosses. Also, I recommend that you watch our videos about statistical analysis and graphical data representation.